your presentation.
Shantanu, it's fine. Mail করলাম এক্ষুনি মেল করলাম হোয়াটসঅ্যাপ করলাম Dr. Shantanu, can we start?
Sir, yes, sir. Give me 1 second. Hello, am I audible?
Yes, yes. Please
Okay.
go ahead.
Good afternoon to everyone. Myself, Dr. Shantanu Ghosh. I am assistant professor at the Department of Pharmaceutical Technology, JISU University. So today we are at the second session, and this is the fourth lecture given by our respected speakers earlier. So today, now with us, Dr. Castro, and I would like to introduce him first with few words. He got his MSc and PhD in chemistry, and then he did his postdoc in 1996 to 1998 in Massachusetts Institute of Technology, MIT, then Tufts University. Later, he joined as an adjunct professor in the Department of Biomedical Engineering, Tufts University in 2001 to 2006, Boston, USA. Presently, he's a top scientist among the Natural Research Council and PI of Nanobiomaterials Laboratory. His research area is obviously the nanotechnology. He published almost 180 papers, different book chapters, 2 books, and 4 patents to his credits. Not only the nanobiotechnology, but biopolymers, biocatalyst, tissue engineering, 3D bioprinting, and pigments are his research interest. So without wasting any time, I would like to give the stage to Dr. Castro. Sir, please.
Good afternoon in India, good morning in Argentina. So thank you very much for your kind invitation. I know my first name is very complicated to read with you. It's Guillermo, with the translation is William. So it's a pleasure for me to be with you. And thank you for Dr. Halder to invite me to this meeting because I have close relationships with many Indian friends since a long time ago, and for me it's a real pleasure. So I will talk about biocarriers. Do you see my presentation right now?
Yes, sir, it's visible. Please go ahead.
Good. So I will introduce one of the topics that we are using in our work at the lab of Nano Nano Biomaterials Lab in Buenos Aires. Right now, I'm moving to Max Planck Laboratory in Rosario, which is associated with the Max Planck Foundation in Germany. So my point at this time is to talk about biopolymers. Why biopolymers? Because we are in our world, we are about 7,000 million people, and we are growing very fast. Right now, we have about 998 million tons of agricultural waste, approximately 80% of the organic waste. So the composition of organic waste, one of the most important are biopolymers. So what can we do with biopolymers? So we just collect the biopolymers. So we use agricultural waste to cultivate with different strains, microbial strains, to produce biopolymers, or right now to extract biopolymers from the waste. We know that the cost of just destruction of the production is very, very high. However, if the application is relevant for the economy, we can do many things like tissue engineering, drug delivery, food processing, and other kind of applications with films, different kind of. So, as an example, nature produced many polymers, just cellulose, chitin and chitosan, wire gum, silk, starch from plants, crustaceans, silkworm and spiders. So most of them just regard silk, in some cases, are wasted. Other ways, we can make a fermentation, and we produce different kind of polymers such as alginate, cellulose, glucans, polydroxybutyrates or alkanoates, and polylactic glycolic acid. These are just examples that we can use these polymers for control release, and this could be very, very interesting. So hydrocolloids are very, very interesting because they are easy to manage, easy to derivatize, and one of the things is, for example, alginate, which is a biopolymer approved by FDA and EMA, just from regulatory agencies of Europe and United States. Also, pectins are waste for plants and is a structural polymer composed of galacturonic acid, just derivatized in different ways according to the plants. We have carrageenans, we have wire gum, we have cellulose, which is a huge waste everywhere. So with this polymer system, we can create different kind of things. So micro nanoparticles, films, microtubes, hybrid nanoparticles and films, and hybrid films. Why we need to create this? Because nature is always producing these biopolymers, or bugs are producing these biopolymers just for different uses. In our case, we need to adapt these biopolymers for our purposes. So we require to produce different kind of hybrid compounds. So right now we are dealing with nanotechnologies. Nanotechnologies just is the top of the wave. Every single paper that contains the word nano is more easily published than the other ones. However, in some cases, nano devices are not required. Why nano devices are the top? Well, first, they comprise a limited area of distance. So we have from 5 to 100 nanometers. Why we have this? Because they was ruled by the physicists, and the physicists believe that the quantum mechanics depends on the size. However, the cells, they don't care. So sometimes it's not useful to use nano devices, just to use micro devices. The best case for that is long administration. Right now, we have a serious problem with the pandemic. And if we want to administrate some drugs directly to the lung, we cannot use nanoparticles. We need to use micro devices. Why? Because if the diameter of the particle is less than 1 micrometer, they will go to the capillary system. But if it is higher than 5 micrometers, the macrophage will be in charge of that, so they will just disappear from the bloodstream. So these are examples of biopolymer organic system. As you can see, they use different kind of polymers, and they are other kind of substances. In most of the cases are other kind of polymers and used for different applications. Most of them are related to biomedicine, advanced biomedicine. Why we need to require these systems for example, for tissue engineering or drug delivery? Well, right now the pharmaceutical industry is the technology of pharmaceutical industry is just very, very old. The technology is dated in 1950s. So we require to have a delivery system that they don't have the problems that currently we have. So we have no just the WHO, the World Health Organization, declare emergency, for example, for antibiotics. And this is why we need to have special systems that contains 1 or more 2 drugs, and those systems can be used for control release. So with defined concentration and time release in the proper place. So these systems wants to be in that area. But not only biopolymers and organic systems are right now emerging from the pharmaceutical technology. Also, we have biopolymers and inorganic hybrid systems. So in this case, we just add different kind of structures to increase the strength, or right now to increase the biocide activity. So such as the case of silver phosphate, for example. So what we can do with a hybrid polymer, for example, lipid nanostructure. So co-delivery, more than 1 molecule that contain the activity. We can produce responsive nanoparticles. We can make stealth surfaces, which is pegylation or covered with albumin or proteins that don't give any immune resistance. We can change the routes of administration according to the structure, and also, and which is one of the advantages, to target the drug delivery. So many applications and many ways to produce a system that can be used for advanced drug delivery. So what we have now? What kind of system we have? Well, we have zero materials. So this is a metallic one. Quantum dots in general are very toxic and used only not for drug delivery, just to create images of tumors, for example. 1D materials, that the nanotubes, 2D, just films and other kind of particles with different kind of sizes. So from dendrimers, which are synthetic, is a synthetic polymerization process, but also very old technologies dated on the 60s, just liposomes, hydrogels, micelles, mesoporous nanoparticles, and polymeric nanoparticles. So what are the main methodologies that we use to produce this? So natural or modified biopolymers to create blends, to coat, make coatings, and hybrid systems, as I mentioned before. The most popular are ionic gelation, in the case that the polymer contain carboxylate ions, precipitation, supercritical fluids, which is a new technology, but is not very easy to manage. So right now it's a challenge to produce a large scale nanoparticles with supercritical fluids, ultrasounds, extraction, just a 3D bioprinter, and microbial cultures. So those are the main seated procedures to produce drug delivery systems and hybrid. What kind of studies we require to do for the approval of a drug delivery system? Well, spectroscopic to see the interaction between different kind of components of the matrix, microscopic kinetics, rheological stability, is math modeling, finally. So all these specialties require a team. No one can do all of them. So this is a new perspective of science. We need to establish a strong collaboration with good people just to produce good products based on different specialties. And this is a new way to work in science, because we work from the 90s in the one science was the top, and right now we are just special. Each laboratory has special things and special knowledge and need to collaborate with others. So I will present 3 cases of biopolymers for bacterial infections. So in cystic fibrosis, we have infected lungs. Cystic fibrosis is an illness that contain a deficiency in chlorine transporter. So they create mucus in the lungs, so the people cannot breathe. So this is a genetic disorder. The kids, because it's a genetic one and starts very early in our life, the kids cannot breathe. So, but not only affect the lungs, also the intestine. And the main bacteria to produce this is Pseudomonas aeruginosa. Pseudomonas is a very complex bacteria. It's very common, it's everywhere, and they can live in all kinds of environments. Since that, the resistance mechanism of Pseudomonas is very hard. So not only survive, but also Pseudomonas produce biofilms over the surfaces. It means that the lungs is totally covered by a mix of alginate and DNA product of bacterial lysis. So the idea is to give a system, inhalation system for the lungs, in order to kill the bacteria. So I mentioned before that microparticles for lungs should be in the range of 1 micrometer to 5 micrometers. So what are the most used antibiotics? Tobramycin and quinolones. But all of them are very toxic. So they require very high concentration because the biofilm in the lungs don't allow to the antibiotics to reach the bacteria. So it's very complex. So we was thinking at that time to use hydrolytic enzymes for mucus degradation. Right now, DNA can be found in the market, but it's very, very expensive. And also, they need to complement these DNAs with an antibiotic. So we were thinking just to reduce the amount of the antibiotic using natural compounds. Natural compounds that contains that are bioactives, just as example, monoterpenes and essential oils, but also to administrate metallic complexes, because we know India is well known because of the silver, of use the silvers, so as antimicrobial. So we think that that could be a good choices. So we produce some particles, and as you can see in green here, this is nanoparticles, and this is lung tissue of a rat. So this is Pseudomonas aeruginosa alive, and this is what happened when we administrate the microparticles. So I will introduce 3 cases: hybrid biopolymers, polymers, synthetic polymers, and biopolymer coating. All these 3 things are just an example of how we can play with these biopolymers. So in one case, the hybrid system biopolymer, we just create one with alginate lias, because Pseudomonas produce alginate, as lipoploxacin, which is a fluoroquinolone, to work together and enhance the antimicrobial activity. So this is lipoploxacin. As you can see, they have sorry, they have aromatic rings, means the structure of this molecule is planar. So what means that the structure is planar? It means that the molecule is very hydrophobic, because they don't have the only one is this one is a carboxylate residue, but in most of the cases this structure allow to piling molecules of lipoploxacin in physiological media, because they contain high oxalate concentration. So sodium chloride in physiological condition is about 154 millimolar. So the administration of free lipoploxacin can cause tendon problems, nerve damage, changes in the mood of behavior, and low blood sugar in the current. So the potential symptoms of administration is headache, burning, pain, confusion, agitation, paranoia. So it's complicated molecule. So in this case, we just found alginate liase. Alginate liase can be produced by several microorganisms and is able to degrade the biofilm produced by Pseudomonas aeruginosa. So this molecule is about 32 kilodaltons, isoelectric 0.8, contain alpha helix and beta sheet, pH 7.5 as maximum activity and 40 as maximal temperature of activity, which is good because our temperature is about 37, a little bit lower, and the specificity is polyhyaluronic acid. So we can just produce hybrid gels with pectin. So pectin has is a polygalacturonate polymer containing carboxylate residues, and in some cases, these carboxylate residues are esterified to produce hydrophobic areas. So the carboxylate residues can cross-link with multivalent cations. In most of the cases is calcium, but could be zinc and others that contains biological that are safe from the point of view of biological life. So, but hydrophobic residues are good because molecules can be attached to them. And I mentioned lipoploxacin contains is a very hydrophobic molecule. So we use alginate, and alginate is composed of mannuronic and hyaluronic acid. They can be cross-linking with calcium, creating the typical X-box structure. So in such case, this is a polar environment. They can interact with pectin, and we was thinking about that, and create an hybrid structure that it will be more disorganized, but can allow to keep inside the lipoploxacin. So there are many techniques to create this hybrid polymers. So this is coaxial flow, which is the more easy, and they will be very easy to scale up. Also electrostatic, but just require high power source, vibrational, just produce a different kind of structure of jet cutter with high speed driver. So we can produce different kind of size and structures, and those are surfaces of different kind. This is a cut. This is a surface of the microsphere, and this is the interior, which have different concentration of pectins and alginates. So, what is the point of use enzymes? So we can do two things. One is a structural modification, just by loading enzyme, and the other is just tailoring the surface of the microsphere, of the nanosphere, whatever is the structure. So there are two applications, two strategies that in both cases are good. So alginate liase, hydrolyze alginate, and produce two different molecules, and unsaturated oligosaccharides, and this one that contains a double link that can react in aqueous phase. So the polymer will be totally disrupted, and later will release the content of the nanoparticle. So those are the structures of alginate. as you can see, the surface is very homogeneous, and the distribution analysis of the distribution is very symmetric. When we make a mixture of with pectin, we create just this microparticle with like a brain structure. No? You can see the folding here of different kind of interaction between alginate and pectin. And when we study the release of lipoploxacin, we can see that the presence of alginate liase in the structure of the microparticle change the release. So in sorry, in both cases, they enhance the release in the first, I will say, 1 hour and a half. So this is very interesting because in this case, we can regulate the release of lipoploxacin, just changing the alginate liase concentration. When we study the hardness of the matrix, so fresh, they have a 0.55 newtons, but when we add pectin, change a little bit. In this case, there are almost no differences between both. So it means that the structure of the hybrid polymers is consistent. So there are no alterations of the structure. This is very important because when we administrate a nanoparticle or a microparticle, they will go to the air, and in this case, because we are making an inhalation system, and they can have a very, very hard time against the fluids of the lungs, and the deposition of that. We can create a different kind of structure that are not good if the hardness change with the hybrid structure. So also with alginate liase, there are no changes at all. When we use trees buffer saline, they diminish in 2 hours. They diminish the hardness, and which is good because in this case the structure of the microparticle just try to dissolve. Also, we see the effect of PBS. In this case, phosphate can be complex calcium ions of the alginate structure, so they thus disrupt the cross-linking of calcium and they reduce about 30% the hardness of the particles. So as you can see, incubation changed the hardness, but also when we add liase, alginate liase is a little bit reduced. The same way in the hybrid systems. So when we use DCC, so calorimetric analysis show changes in the structure, which is correct, but it's not too much changes. It means that the structure, as we mentioned before, analyzed by the hardness, is very close. So this confirmed that the good structure of the hybrid system. When we study the swelling at pH 7.4 and 37 degrees, we can see that addition of alginate liase change the swelling of the particles. So it means that alginate liase work. But the interesting thing is we use alginate liase in a microparticle, included in a microparticle that contains alginate and pectin. So if we produce this, we need to inactivate the enzyme, because if not, we have no particles at all. We will have some, only pectins and some residues of alginate. So in this case, we just study what are the pH in which the alginate liase is inactivated, but not more than inactivated. I'm sorry, is inhibited, but not inactivated. So at pH 4, the enzyme, it doesn't work, but we can enclose the enzyme in the structure containing alginate and pectin. However, when we switch from pH 4 to pH 7.4, alginate is active and start to work. So start to release the cargo, which is first lipoploxacin, and second, the enzyme by itself, because it will be active on the surface of the biofilm. So when we incubate in PBS, the structure will be like a donut. You can see this was the picture before of the microparticle analyzed by TEM. This is the donut that I mentioned. So it means that the alginate liase is inside the microparticle, as we showed before by swelling, but also in the interior of the microparticle. So the diffusion will be go from inside to outside, which is advantage, because in this case we can control it by the amount of enzyme, the kinetic of microparticle degradation. So this is after 2 hours, but what happened later? And later on, we have nothing. So the microparticle was totally destroyed. And this is advantages, because if we remember that the lungs will be covered by a biofilm. So we want to destroy a biofilm, and we don't want to add more hydrogels to the lungs, because in this case we will be worse for the patient. So when you use a system contain calcium carbonate, which is an hybrid system, and we add alginate and lipoploxacin plus alginate liase. As you can see, control bacteria this is bacterial survival. You can see the effect in the presence of alginate liase. So the survival is very, very low. It's about 2%, just in 6 hours. So this is the particles just put in a plate containing Pseudomonas aeruginosa, and you can see what happened in the presence of alginate liase. So there are no microsphere. In the case that we don't have alginate liase, the structure remains. That this was mentioned before. So what happened for oral administration? Oral administration is very complex either, because they require to go. It's more easy for the patient because they don't require any kind of technologies as a receipt with the antibiotic. But also from the point of view of drug delivery, it's very, very complicated, because they go across the stomach with a pH 1.2. So we want to preserve the activity of the antibiotic and also the enzyme in this case. So a pH 1.2, when we put only alginate, is totally hydrolyzed. However, when we add pectin to the structure, as you can see, we have in 2 hours, we have a difference between 40% and about 90%. So it will be something like 50% difference, means that we can control the release by two ways. As I mentioned before, to change the ratio between the two polymers, so alginate and pectin, and also with the presence of alginate lias, which is this advantage because we can, in some ways, to make the structure required by the characteristic of the patient. A pH 7.4 is also the same kind of trend. So the presence of alginate lias change the profile as well, the pectin. So this is very interesting because we can play in different alternatives. Also using different kind of alginates and different kind of polymers of pectin. Sorry. So the length of the polymer will be can be changed by different kind of hydrolytic system to create different lower molecular weight polymers or larger by just an enzyme reaction. So we need to also to test what happened with the toxicity of the cell. So with a different kind of concentration of antibiotic concentration, and we test the viability of 3T3 NIH cells, and we can see that no effect of toxicity of the empty matrix, just with alginate or in presence of phosphate or control. So it means that not only the matrix is innocuous, but also the products of hydrolysis is too. So we go later, we include the antibiotic, and as you can see, at 18.2 mg per ml of the lipoploxacin, the toxicity is higher. So for chronic illnesses like cystic fibrosis, this can create a serious problem. So in other cases, the toxicity doesn't exist, or it is very lower. So we will introduce another hybrid matrix, which is a biopolymer synthetic polymer. So in this case, we can use a florfenicol, which is a very toxic drug, and they require it's used in animals. It's an antimicrobial in animals. They require to administer it several times a day, which is a real problem for animals. We use alginate in this case and eucradite as for modulated pH. So this is the drug. This is a synthetic mechanism. Is similar to chloramphenicol. So the inhibition is protein synthesis in the ribosome, uses for respiratory diseases, mainly in pigs and bovines. So imagine if you need to administer pigs and bovines with a florfenicol many times a day, it will be a huge problem. So the drug is poor soluble, require, as mentioned, daily doses, high toxicity for liver, kidney, testicle, and this is a serious problem for reproduction, immunosuppressive, and anemic effect. So it's nasty. It's a very nasty drug. So we want to encapsulate this in Euradit reas, which is a cationic one. It's a synthetic polyacrylate polymer authorized by many regulatory agencies like FDA and EMA. They contain a nitrogen and amine group that can create positive charge. So in this case, combination with alginate that contains carboxylate ions can create a network that we can manage for the release of the antibiotic. So we use different kind of three concentrations. As you can see, the amount of encapsulation enhance when we increase the concentration of the antibiotic. So it means that in some cases, the antibiotic is just precipitated inside, which could be a serious problem because the heat of the solution in general is very high. So also we have a problem when we use alginate beads. So the pore size is in this range, and the molecule is this. So it means if we include at the beginning the fluorphenicol inside the alginate matrix, it's very clear that the molecule will be diffusing through the structure of the sphere. So it's at least several thousand times lower than the pore diameter. So we need to have another kind of system to in some way stop the release of or control the release of the fluorphenicol. So those are the structures of a different concentration of fluorphenicols, and as you can see, they start at 5 minutes, and at 20 minutes, the growth of the microparticles are very good. So when we study the structure of the spheres, we can see that when we increase the amount of fluorphenicol, there is a change of the structure. So the size of the particle will be a Fisher distribution compared to the Gaussian distribution. So what is the advantage? Well, the particle size will be lower, but it's not symmetric in this case at higher concentration. So we can just study the release at different concentration of both systems, and there is a difference between both. So at a lower concentration, the release is faster compared to a lower one. This is a pH 1.2. The same things happens at pH 7.4. So this is a complex structure. So we add Euradit. So we keep alginate at 2%, and we add different concentration of Euradit. As you can see, addition of different concentrations enhance the loading of the fluorphenicol. But also there is a increase of the diameter of the part. But in this case, there is no problem to increase the diameter of the particle because it's oral administration. So it's easy to do it. And also we keep one concentration we select based on previous results, the concentration of fluorphenicol. Sorry. As you can see, this is the fluorophenical release with different concentration. The profiles are very similar at pH 1.2, but at 15 eurodid concentration is lower. It means the interaction of the fluorophenical with eurodid is maximized in this condition. So also pH 7.4, it means that we can change the release, as we mentioned before with pectin, just by changing the eurodid concentration. And this is in phosphate as well. Similar situation. In this case, as you can see, is first time is lower at eurodid 50. We want also to analyze the interaction between the compounds. So as you can see, fluorophenical has many peaks, but when we encapsulate in the blend, there is a reduction of the structure. So it means that some of the interactions, some of the structure of the fluorophenical is interacting with alginate and with eurodid, mostly with eurodid, which is good. So also we use DSC, and as you can see, there are changes of the structure of the fluorophenical in the blends. So when we analyze the X-ray by X-ray technique, the structure, this is pure fluorophenical. This is in the mixture. It means that blend structure. And you can see the crystalline structure is very good. As I mentioned before, when a drug crystallizes inside the structure, is very difficult to dissolve, and in such case, the energy required is very high. So in some cases, this is not possible and can create serious physiological problems for the person. So look at what happened with the blend. So there are no crystalline structures at all. And this is very, very important. It means the drug is inside the particles in amorphous structure. So when we study the morphology, so there are no significant changes on the morphology, but when we analyze deeply the surface, we can see that when we add alginate, but with eurodid 15%, the structure of the surface is more homogeneous. Finally, we study the Staphylococcus aureus inhibition with different formulation. As you can see, in 24 hours, there is no growth of Staphylococcus aureus, the same as in E. coli. Well, the third one, part of the presentation, I think it's not too late. It's not too late. Or it's later. Please let me know.
No, sir. Sir, it's okay. You can proceed.
Sir, please go ahead, sir.
Okay, thank you. Thank you.
It's fine. It's fine.
Thank you. So the third case is hybrid system contain always oflofloxacin, which is another kind of fluoroquinolone with eugenol. Eugenol is a natural compound that have some interesting biological activities, and we load in solid lipid nanoparticles with targetable antimicrobial properties. So this is ofloxacin and this eugenol can enhance drug skin penetration, and is importantly is an inhibitory of quorum sensing at so inhibitory concentration, so it's a very low concentration. Eugenol also is liquid. It means that we can mix with other kind of lipids to create a solid lipid nanoparticles, which is very interesting. Also, because ofloxacin is a very hydrophobic molecule, in oil like eugenol, we will more dissolve. So this is a synergic activity, means that eugenol can interfere with the quorum sensing, and ofloxacin can be reduced the concentration in the formulation. Sorry. So we can create two types of, this is more general, and there are at least 4 types of solid lipid nanoparticles, but there are 2 main solid lipid nanoparticles. One is the common one, this that contains one type of lipid, and in this case, a surfactant, but contains also styrolate with another polymer. And the second one is a mix of different kind of lipids. One is solid and another is liquid, also with a surfactant, but decorated with chitosan. Chitosan is the only natural cationic polymer available in the nature right now. So this is an advantage because it's mucoadhesive. But not only is mucoadhesive, but also interferes with the membranes of bacteria. So we have, in these cases, we have mucoadhesion, interference with the transport of components in bacteria, and in the presence of eugenol and ofloxacin, we have a combo that we can reduce the concentration of the antibiotic, which is an advantage. So the mixed formulation increased almost 80%, and the PDI also too, because they contain solid lipid nanoparticle, chitosan, eugenol, and ofloxacin. So the encapsulation diminished, but is good enough from the pharmaceutical point of view. Only is the minimal amount of antibiotic required to encapsulate is 20%. And in this case, we have no problem because we want to reduce the concentration of the antibiotic for administration to reduce secondary effects. So we choose this structure, and we analyze this. So this is microscopy, and as you can see, those are the lipid structures on the nanoparticles, which is very homogeneous. The release is in this way in 24 hours. As you can see, the complex solid lipid nanoparticles, floxacin, is better than the other one. So this is free one, and this is the other formulation. So it's interesting because also we can regulate, based on the composition of the nanoparticle, we can regulate the release. So when we analyze X-ray, so this is again the structure of floxacin. This is a crystalline structure, and as you can see in the solid lipid nanoparticles, there is a reduction of the crystalline structure, which is good. FTIR also shows some interactions between the molecules. So we can see that it's just not to physical interaction, but this is a chemical interaction with different kind of groups. So this is the effect of the formulation. As you can see, in Pseudomonas aeruginosa, the concentration of the floxacin is reduced and reaching low effect also in Pseudomonas and Staphylococcus aureus. When we analyze the formulation at 4 hours with positive and negative controls, means that the cells are alive or dead, we can see this this situation. This is just with eugenol without antibiotic, but when we add ofloxacin, we can compare, and the difference are very high. Means the efficiency of the system with low concentration is better. So this is optical microscopy, and this is fluorescence, and this is TEM, transmission electron microscope, and you can see the bacterial cells are totally destroyed. So this is Staphylococcus aureus, and you can see the debris here. It means that the cells are killed. So when we choose cultural cells, this is epithelial cells, and we see the cell viability. In the cases of the formulation is good. There are no toxicity for the solid lipid. So the cell viability is good with different concentrations. At high concentration could be toxic, but not at the concentration required for a treatment. So finally, we can manage the polymers to create a different kind of structures. There are novel candidates for drug delivery, and we can create many hybrid systems. We can tailor this. We can cut in a short molecular weight. So we have many tools to modify right now biopolymers for drug delivery. So we can create many efficient matrices and advanced for drug delivery in a simple way. The role of biopolymers can be also defined according to each device and target, and can provide structural and chemical specific properties that can be easily tailored. So this are the works in my lab, just the research areas and lines that they are doing many of my students. This is my place, and this is the staff of my work. This are the collaborators. As I mentioned before, we require to have different specialties. These specialties strong the research. So I know some friends that Arup Mukherjee is in the conference too. He's a close friend of mine since long time ago. Ashok Pandey, Ashok Nada, among the most close friends. So this is my lab. Thank you very much. This is my publication of this year, some of them. Thank you very much for your time. I'm patient with my bad English. So I remain at your disposal.
Yeah, of course, there will be some questions, sir. In fact, it's indeed a complete presentation and very good, audible, legible English.
Thank you. Thank you very much.
Yeah, Dr. Austin, please.
Yes, thank you so much. Really, it was interesting, and we learned a lot. That is the main purpose of this faculty development program. So we are here to learn something from you, sir. I think that 100% we are satisfied, and that help our participant to do something, to think in that way, to develop some product. So always your works inspired me actually personally. I do really follow your article and try to replicate something in our lab with the limited facility we have. I think the similar activity will be doing by our participant. They today they have seen many varieties of work, different dimensions of applications of biopolymers, and that will be really helpful to them. So any questions, open questions from our participant we are expecting, you can directly talk to our guest, and sir will be very happy. I know sir is very cooperative since I met him, and sir will be very happy to answer your questions. So any participant. So before that, sir, I will take 2 minutes, ask you some questions. So initially, sure. Yeah, thank you, sir. Initially, you talked in your presentation that synchrotron facility. So that yes. is available. Sir, what is that actually? What kind of activity or the facilities which are available can be accessed there?
Well, there are many synchrotrons right now. We use synchrotron in France. So France has open windows for grant proposals. So you can send a proposal to the synchrotron parties. They will evaluate you, give you some time to analyze your samples. Also, there is in my case, not for you, because France is more close, but in Brazil we have 2 synchrotrons. One is a biosynchrotron, which is used to analyze different spectroscopic ways cells and the effect of drugs in cells, components of the cells. So an interaction is in very fine conditions, because, for example, IR is about 10,000 more sensitive in synchrotron than the tabletop FTIR. So it's more sensitive, you can get better results. In Brazil also, they have the similar system as in France. You can send a proposal and send the samples, and at the starting point, they will analyze your samples for free. Later on, they will arrange with you the conditions, but it's straightforward.
Okay, sir. Thank you. So another thing, when I was listening your lecture, you said some alginate gels, something was prepared, developed, and you have performed some characterizations in FTIR. So can you elaborate more on how to, because in FTIR, we basically use powders, we dry it, then we mix with the KBR, then actually we punch it and use for the IR characterizations. But in case of gel, when you're handling gel, how it's possible? Is it possible by FTIR normal basic instrument?
Well, yes, it's possible, but you need to dry the gel. In this case, we use ATR technique for analysis, which is the sensitivity is not very good as the other one, but we can find some displacement of the main peaks, and we can check the water content and how the water content is associated, and also as well the position of carboxylate or carbonyl groups are very straightforward to analyze, and amine groups too. So we can see how are displaced these peaks. Based on that, we can say that there is a kind of repulsion or if it is going to the violet area or blue area, or it's going to the red one. So it will be a tool to realize how interact, for example, a drug with another compound, just a polymer. But of course, they need to confirm this by other ways. For example, calorimetry. So calorimetry will show the degradation with the ramp, with the temperature ramp, will give you an idea how the interaction is between the polymer and the drug or different kind of molecules. So, but there are no one single tool to analyze the system. We need to combine this in many ways.
Yeah, definitely. Yes.
Sir, in this context, may I ask one question?
Sure.
Yeah, sir, in the FTIR spectra, the interaction that you have studied, what I can see, that exactly you have measured the position of carbonyl group. When you have studied the interaction of chlorphenicol with alginate, is there any shifting of this carbonyl functionalities?
Yes.
Or
yes.
what exactly you are measuring?
Well, we measure it as when we use IR, we use ATR. ATR is not very sensitive, and also IR is not very sensitive too.
Right, sir.
So we just give an idea. We don't try to use a quantitative analysis, just qualitative. But we, yeah, but we can see, for example, how a band, a carbonyl band, is placed from one point to another. If going to the violet area, it means that the interaction of this polymer with the molecule is not good.
Right.
But if you're going to the red one, it's on the contrary. So there are kind of association, but also we need to analyze by X-ray. If it is possible Raman, if it is possible MNR, if it is possible. So they need to combine techniques. The
so
conclusion is,
yeah, sir, only FTIR does not suffice, right, sir?
Not only IR, X-ray also, or DCC is also. So this is why we need to use several tools and combine the tools to give a conclusion. Because one technique could give you some ideas, but they need to confirm by other kind of techniques. So there are no there is
conclusive technique, right?
Yeah, it's not only one. So the heaven is not by one technique. You need to combine them, and also combine with modeling, with release. So there are many toxicity. So many things to need to do. This is why we need to work in groups. So teams with different expertise, because it's not possible to have all of them in one single person.
Sure. Thank you, sir.
Thanks to you.
Yeah. So I think there is no more questions in the chat box. I think we should move on. And indeed, it was a very wonderful session, sir. Thank you once again. And
thanks to you. It's a pleasure for me to talk with you, and I remain at your disposal. So there are no problem. You know my email. Everybody can contact me, and I will help as much as I can. Of course, I have also limitation, as I mentioned. So it's something that it's good for us to collaborate and to see different perspective of work. Thank you very much for the invitation, and it's a pleasure for me to.
We are
Okay,
so happy,
be with
sir.
you.
We are so happy that you are here with us. So,
Okay.
with this note, in this pandemic situation, let us share a small token of appreciation from all of us, sir. Kindly accept this one. It's
Oh, thank you very much.
sir,
Thank
we'll
you.
mail it to you also. Thank you, sir.
Thanks to you.
Thank
Be
you.
safe. My best wishes for you and your country, your lovely country.
Thanks, sir. Thanks. So, with this, we are concluding, sir. Thank you.
Thank you very much.
So, dear participants, we have come to an end of day 3, session 2. We will close here, and as usual, we will have an Google form based feedback